So the next speaker is Florian Wünneman. He's an expert in the field of cardiovascular disease and functional genomics. He's currently a postdoctoral researcher within the team of Dennis Shapiro at the Institute of Computational Biomedicine in Heidelberg. Florian completed his PhD at the University of Münster in collaboration with Dr. Hendelfingers at the CHU Saint-Justine in Montreal, where his research primarily centered around congenital heart disease. After obtaining his PhD, Florian joined the research group of Guillaume Lettre at the Montreal Heart Institute. There, he focused on the genetic architecture of complex cardiovascular diseases using functional genomics tools such as pooled CRISPR screens. The title of his presentation is Using CRISPR Perturbation to Prioritize Coronary Artery Disease Risk Loci in Vascular Endothelial Cells. Florian, whenever you're ready.
Yeah, thank you so much, Jan. First of all, thanks so much for the invitation and giving me the opportunity to talk here. I think, like John was saying in his talk, many of my friends have presented here, and I'm really honored to be able to present. I'm happy to share one of my papers with Guillaume during my postdoc with him about using CRISPR perturbations in vascular endothelial cells to probe coronary artery disease loci. And I want to start by just talking quickly about what is coronary artery disease and why we're interested in this. So coronary artery disease is basically the buildup of plaque within our coronary arteries, which can ultimately lead to heart attacks. And this is a process that happens in all of us with age, but there are certainly specific things that increase our risk of developing this. And when we get this buildup, we also call this atherosclerosis. And so, as I mentioned, there are many risk factors for atherosclerosis. Some of them are immutable, like your age, your sex, your ethnicity. Some of them are like alcohol, smoking, diet, and physical activity. We can modulate ourselves in our environment. And some are predisposed, like having diabetes, dyslipidemia, being obese, having hypertension. But the one that we want to focus on our screen and that I want to talk about today is obviously genetics. And from the genetic side, there have actually been many risk loci already identified for coronary artery disease. This figure is actually quite old, 2017, but it's still like my favorite one because it sums up the different pathways that coronary artery disease acts by quite nicely. I think someone should make an updated version of this. And we can see that there's lots of different pathways of how risk of coronary artery disease is conferred. For example, LDL cholesterol, like the best known one, I think, but also insulin resistance, inflammation, cell adhesion, and other pathways contribute to the risk of coronary artery disease. Now, when we designed the screen, this was in 2018, that are associated with coronary artery disease. So at this time, we actually took 92 variants, sentinel variants that we used for our screen. But I want to obviously highlight that now in 2023, there's way more studies that looked at genome association studies that looked at coronary artery disease. And now there was a paper just recently at the end of 2022 that actually identified more than 250,000 variant associations. So just to put that in the context of when the screen was created. So for our screen, the way that we did this is we took these 92 sentinel variants and we looked at linkage disequilibrium and took variants that are in kind of strong linkage disequilibrium to those sentinel variants to define the genomic regions that we wanted to look at. And within these regions, for each of those sentinel and proxy SNPs, we then tried to find the 5 best single guide RNAs for each of those SNPs, which gave us a total set of about 7,400 guide RNAs. And we took those guide RNAs that are basically targeting different SNPs in the genome. And by targeting, I mean they're as close to the SNP as they can be. Not all of these guide RNAs, of course, overlap the SNP because the PAM sites sometimes aren't as close to the SNP as they could possibly be. And then we also included some other RNAs, guide RNAs in our screen, some that are targeting essential genes to make sure that our screen works, some that are targeting known genes that are involved in some of those pathways that I've shown, and some positive promoters, and you'll see later why we need promoters, that are also targeting genes, promoters of genes that are known to be involved in pathways of the cellular system that we care about. And that using all of these guide RNAs, we ended up with a final guide RNA library of 8,051. Now, these guide RNAs, for most of those SNPs, we're actually able to design 5 guide RNAs, but of course, for some of those SNPs, there's just not enough PEM sites within the window that we defined, or the guide RNAs that we could identify did not pass the quality control thresholds that we set to have a very high targeting library. Now, when we look at the average distance of guide RNAs from the SNP sites that we're targeting, this is around 23, and you can see that it's kind of like almost normal distributed. So we kind of limited ourselves to saying we want to go not too far away from the SNPs that we're targeting, and so this is a maximum of 50 base pair windows in both directions, so 100 base pair window around the SNP. When we look then on how many proxies within association locus we can actually target, you can see that for some sentinel SNPs, we can actually target all proxy SNPs within this locus using guide RNA. But there are also significant, like loci where we cannot target, where we can target less than 50%, for example. And this will obviously limit our ability to interpret the findings within these loci. And not surprisingly, but we still want to show this, is that most of the variants that we actually selected are intronic variants. As we know, most of the associated variation for complex traits like coronary artery disease actually falls within intronic regions. Now, for coronary artery disease, the reason why we wanted to do these screens in the first place is that for most of those variants that we know are associated with coronary artery disease, we actually don't know what's the biological function and what are the genes they're acting through. So for about 50%, this number might have changed a bit since these new studies came out, but for about 50%, we don't really know which genes and which pathways these variants are acting through. And we think that some of those 50% of unknown variants are acting via endothelial cells, which endothelial cells are forming basically the inner lining of the coronary arteries and are extremely important for the intactness of the coronary artery. And they're also directly involved in the disease process of coronary artery disease. And they are involved in this mainly, there's lots of pathways, but mainly by expressing these adhesion molecules, so E-selectin, ICAM, VCAM, PCAM1, and these are kind of involved in facilitating monocyte or immune cell adhesion to the endothelial cells and infiltration via the endothelial cells. But there are also like functional pathways like reactive oxygen species signaling, nitric oxide, and calcium signaling that play really important roles in endothelial cell biology. And so we wanted to take these endothelial cell properties and use them in our screen to probe how endothelial cell function might be involved in conferring risk for coronary artery disease. Now, to be able to use pooled CRISPR screens in vascular endothelial cells, we had to engineer vascular endothelial cells that readily express these different CRISPR variants that we wanted to use. We chose to use 3 different CRISPR variants in our screen. The first one is CRISPR-Cas9, the one that everybody knows, where we can use guide RNAs to induce indels at specific genomic regions. We also use CRISPR activation, where the Cas9 is inactive and doesn't actually cut anymore, but it still binds and has an activating domain linked to it, so that when we guide it to a specific genomic location, it should actually activate gene expression at that locus. And we also use CRISPR-A, where we have a repressive domain linked to the Cas9. And so instead of activating, now we're blocking gene expression in a specific genomic locus. Now, what we then did is we took all those guide RNAs that I've talked about earlier. We made them into different plasmid libraries and we sequenced those to make sure that the distribution of those guide RNAs across the library is more or less even. We then created 4 different independent batches of lentiviruses to make sure that this is a really reproducible process that we're doing. And then we used them to infect these 3 different TLOHEC cell lines, or maybe didn't mention that we use TLOHEC. So TLOHEC is an immortalized human aortic endothelial cell line that is our model system to look at coronary artery disease. So we then use these viruses to infect these cell lines. And for Cas9 as well as CRAB, we stimulate these cell lines using TNF-alpha, which puts these cells in an inflammatory or pro-inflammatory state. And since we are blocking or cutting or inhibiting gene expression, we wanted the cells to be in this inflammatory state and then see what kind of pathways we might be blocking. In contrast to this, when we're activating gene expression, we didn't want those cells to be in an inflammatory context so that we can actually observe potential mechanisms that activate these types of signals. We then fact sort these different cell lines after 7 days after infection and antibiotics selection, and we collect the top 10% and bottom 10% for these 6 different markers. So we look at E-selectin, VCAM1, ICAM1, ROS signaling, nitric oxide, and calcium signaling. And for these, we collect the top 10 and bottom percent of cells, and we sequence the guide RNAs that were found in these cells. And then this enables us to compare the top to the bottom, but also it enables us to compare these different fractions to the guide RNA libraries themselves. And so the first thing that we looked at was kind of treating each of these independent libraries as one sample. So each dot in this plot represents one of these top and bottom 10% libraries. And we wanted to see whether, if we treat them like a sample like RNA-seq or similar to this, whether the signatures of the top 10% of guide RNAs that we find within each sample reflect their different Cas flavors. And this is exactly what we see. Like you can see here, samples that come from CRISPR-I libraries cluster with CRISPR-I libraries, from CRISPR-A with CRISPR-A, and from Cas9 with Cas9, indicating that these different Cas flavors really have distinct effects on these different cell lines and on these different phenotypes. And the next thing that we wanted to look at was using these essential genes as a positive control to make sure that our screen actually works. And what we expect here is that we identify less guide RNAs in FACS sample pool. So we're comparing to the plasmid. And what we would expect is that for Cas9, where we are destroying these essential genes or cutting them and making them inactive, we would expect a shift to the left in these essential gene guide RNAs compared to all the other guide RNAs in the pool. And this is exactly what we see. So this is very recomforting that when we cut essential genes using Cas9, we do find a depletion of these essential genes in Cas9. We find a similar effect in CRISPR-I, less strong. This probably comes from the fact that these are very short genes, these essential genes, and we're basically blocking their transcription by putting CRISPR-I anywhere on these SNPs, on these reading frames of those genes. We also were surprised to see this effect in CRISPR-A. We think that this comes from steric hindrance of the Cas9 moiety in these very short genes, thereby also, at least in these essential gene context, blocking instead of like activating essential gene expression. But we were happy to see that Cas9 and CRISPR-I kind of confirmed that our screen was working. Now, we also included positive controls for these specific readouts that we looked at. And the easiest ones to include, and that what I want to highlight here, are those three adhesion molecules, VCAM, ICAM, and E-selectin. And here's one example of what that looks like. This is the VCAM1 locus. You can see here that these are guide RNAs we derived from existing libraries called Gecko and SAM that lie within on exons of these open reading frames, and also include promoter guide RNAs that would be useful in the context of CRISPR-A. Now, when we look at these positive controls, we first looked at Cas9, and you can see the three different adhesion molecules here, each per column. And what you're looking at is on the x-axis is the rank of where that guide RNA is ranked within sorting all the guide RNAs based on their effect size. And on the y-axis, you can see the beta, so the effect size of that guide RNA within that pool. And what we can see is that these guide RNAs targeting the actual locus that we're measuring, so in this case, E-selectin, really all rank like the highest. So if we cut E-selectin, we're reducing E-selectin and negatively like depleting it in the top 10% and thereby getting a negative beta when we compare the top 10% fraction versus the bottom 10%. We see the same effect of ICAM, but for example, in the ICAM locus, the cutting at the promoter doesn't actually exert a big effect like it does in E-selectin, but still cutting like using Gecko guides on the exon. Now, I expect when we block promoters and we block like early exons of the genes, we find a depletion of these markers, of these guide RNAs targeting the markers that we're measuring. And this is exactly what we see. So here again, we can see that guides targeting selectin, ICAM, and VCAM rank the highest within CRISPR-I. Now for CRISPR-A, we expect exactly the opposite. We expect a positive enrichment of these guide RNAs in the top 10% expressing E-selectin, ICAM, and VCAM, because we are activating the expression of these markers. And again, this is exactly what we see. If we put the guide RNAs at the promoter of these genes or at very early exons, like the SAM-ICAM guides, we see a strong upregulation of these positive markers within our screen. So all of these findings show us that our screen is working as expected, and our readouts work in a way that if we use the potentially strongest guide RNA that we can target to this locus, we find a very strong signal. Now then, we applied this pool of with the actual guide RNAs targeting the cat loci and looked at these results. And we were happy to see that we could actually identify quite a few associations of finding guide RNAs that have strong effects on these different measurements that we find, and that we also identify measurements across the different, or results across the different measurements that we have. So you can see that we can find quite a few different combinations of CRISPR flavors and vascular endothelial cell phenotypes here. In total, we identified 51 variant endothelial phenotype associations. these comprise 42 unique variants that are located within 22 unique CAD loci, meaning that there's some CAD loci that are identified multiple times. So this was quite interesting. Many of these variant associations that we identified were previously known to be associated with endothelial cell function, but some of them weren't. And we wanted to validate some of these findings to see, what's actually the false positive rate of our screen, and if we can find the actual causal gene for some of these regions. So we choose 8 different regions that I've highlighted here: ARNTL, furin FES, DHX38, MAT2A GGCX, and a few others. And what we did is we focused on hits that these 8 hits basically had very strong effect sizes or betas, and were often positive for more than one assay. So they might be positive for Cas9 selectin, but also for Cas9 ICAND, meaning that there's multiple lines of evidence that suggest that they might actually assert effect on vascular endothelial cell health. From these single guide validations, so what we did is we took the strongest guide that we found in the screen and did independent infections of these guides within cell lines and measured the phenotype only using this one guide in the cells using FACS. And doing that, we were able to validate 6 out of 8 hits from those pooled screens. And this kind of looked like this, where for example, for DHX38, we compared these to safe harbor guides. So these are guides that are binding somewhere in the genome where their effect should not in any way affect the cell system that you're looking at. And we derived these from like a paper that had like an extensive list and tested these. So these should not have any effect. They should have a null effect. And what we see for our validations is, for example, that for DHX38, we see the same shift that we saw in the screens. So we're quite happy to see this quite high, at least we thought that it was quite a high validation rate. So then I want to go into some further validations that we did using RNA sequencing of some of these validated single guide RNA infections, which we did to really try to pinpoint what is the causal gene in some of those locus. And the way that we designed these is that we took the candidate guide RNA for the locus, and again took a safe harbor guide RNA. And we only took a Cas9 or CRISPR-A validations because none of those 8 hits that we validated was actually CRAB, the inhibition CRISPR-I. So we took these guide RNAs, we made viruses, we infected these cells, and then for the Cas9 ones, we simulated similar to the screen. We stimulated them with TNF-alpha, selected them using antibiotic selection. But for CRISPR-A, we did not treat them with TNF-alpha, and we also cultured them only for 5 days instead of 7. We then took these 2 different groups of cells and did bulk transcriptomics on them and tried to identify transcriptional responses to the target guide RNAs. Now, the first locus that I want to talk about is the FES-furin locus, which showed a strong signal for E-selectin with CRISPR-A in our screen. Now, here you can see the locus, and you can see that there's 2 genes within this locus, furin and FES. Our sentinel SNP from the GWAS is actually located overlapping the FES ORF. And the candidate locus that SNP that we identified, RS12906-105, is actually located right in the promoter of FES. And you can also see that from public data that there are some attack peaks overlapping FES and furin within endothelial cells. These are like human aortic endothelial cells, so similar to what we have. And that in RNA-seq, both of those genes are also expressed. And even that using like high-C, that the promoter of FES actually interacts with the promoter of furin. So we performed RNA-seq on this locus. And what we found is that when we guide CRISPR-A to this position of the SNP here, we actually find strong upregulation of FES, but also upregulation of furin. When we look at all the genes in cis that are close like to this SNP. So this is kind of confirming that this guide RNA actually works, right? That the gene that's the closest, that we are putting CRISPR-A on the promoter, is actually upregulated very strongly. And as you can see, FES is upregulated a bit stronger than furin. Now, this kind of points towards the fact that, okay, FES is obviously having a stronger upregulation within our screen, or is having this guide RNA has a stronger effect on FES, but it doesn't really highlight whether FES or furin is the causal gene at this locus. So what we did then is that we actually did base editing at this locus, and we swapped the allele at the locus from A to G. And what we then did is we again treated these cells using TNF-alpha and measured expression of both genes with and without TNF-alpha in the context of both of these SNPs. And what we found was really interesting. So when we do this for furin in the context of AG and GG, TNF-alpha causes a reduction of expression of furin, independent of genotype. But when we look at the same effect of FES, in the AG context, we do see a reduction of FES expression. However, this reduction vanishes when we introduce the GG genotype, meaning that this effect of this SNP on FES expression within an inflammatory context is genotype dependent and might actually point towards the fact that FES might be the causal gene at this variant, or that both genes are actually implicated in coronary artery disease risk. Now, the second locus that I want to talk about is DHX38, and it's a bit of a more difficult story that is not as straightforward as FES furin. Now here, the story is a bit more difficult. As I said, we have a sentinel SNP that overlaps the DHX38 ORF, and then we have two SNPs that we found overlapping coding exons of DHX38. So in this context, Cas9 is likely cutting the coding exons of DHX38, causing loss of function of DHX38, and that is probably conferring the effect that we see in our screens. We were also asked during the review process quite extensively whether the risk might be actually conferred by the LDL-C SNPs in this region. And we've done tests to see whether there is independence of these signals. And it seems, at least from our point of view, that it's much more it's quite unlikely that this risk in this locus is actually conferred via LDL-C. So looking at DHX38, we did RNA-seq and compared the safe harbor gene expression to the gene expression of the guide RNA targeting the coding exons of DHX38. And what we found was quite surprising to us is that DHX38 was actually not downregulated, which is what we would expect, right, if we knock out this gene using Cas9. Now, while we did not find downregulation of DHX38, we did find dysregulation of many other genes, most of them involved in TNF-alpha signaling. Since we're treating these cells with TNF-alpha, that might not be surprising, but we're also treating the safe harbor with TNF-alpha. We also found pathways involved in cell cycling and basically cell activation. And this was quite puzzling to us. We also looked at indel formation within these targeted DHX38 libraries, and we could not identify many indels within the DHX38 reading frame, either suggesting that our assay that our guide RNAs weren't working, or that something else was going on. And so we asked ourselves, why do we see the signature of cell cycle arrest and cell activation without actually seeing a direct effect of Cas9 on DHX38? And our hypothesis was actually that DHX38 indels induce a potential paracrine response in non-edited cells. And this comes from the fact that cells that are edited, we believe, proliferate less, do have DHX38 downregulated, but then exert a paracrine effect on the cells around them, which are the ones that we're actually observing in the RNA-seq, and that would have actually the phenotypes that we found in the screen. And now to test this, we derived a new experiment where we replaced the antibiotic selection by introducing a crimson vector within added to our guide RNAs. So now what happens is cells that receive a guide RNA will express crimson and therefore be fluorescent, and cells that did not receive a guide RNA will actually be negative for crimson. So we can then sort these cells so that we end up with cells that have definitely received a guide RNA, which we can test by fluorescent signal. And we can then compare and see, is there a difference in cell proliferation? Do we find DHX38 downregulated as well as senescence and other cell activation markers upregulated? Do we find indels? And we also looked at beta-gal activity as a proxy for cell senescence. Now, the first thing we did was look at the proliferation. And so we looked at the percent of crimson positive telohex cells throughout the time points of culturing these cells after infection. And what we found is that during the first 4 days, we did not find a difference between a safe harbor guide that does not have an effect and a guide targeting DHX38, but we did find a negative effect of DHX38 at day 7, meaning that these cells proliferated less, and therefore we observed less crimson positive cells at day 7. We also interestingly found that we found much more indels as early as day 4. So this seems to be that that these guide RNAs can induce indels using Cas9, but that this effect only really is strongly observable within day 4, and that at day 7 we find these indels and also this proliferation effect. We then wanted to look at DHX38 expression and expression of markers that we observed in the RNA-seq like P21 and other senescence-associated markers. So senescence, as I've mentioned this earlier, is a process that basically is exactly what we saw in these cells, is that cells stop proliferating, they have all these stress markers that are upregulated, and that is a process that is involved in coronary artery disease. And so we were really interested to see whether we can find the signatures that we observed in the RNA-seq also in these crimson experiments. And what we find is exactly that beginning at day 4, when looking at DHX38 guide RNAs, we find that at day 4 and day 7, DHX38 is actually significantly downregulated in guide RNAs targeting DHX38 compared to safe harbor, but also in cells that did actually receive a guide RNA with crimson versus those that did not receive one. And reassuringly, also CDKN1A, which is P21, this senescence marker, is also upregulated at the same time. Finally, we also looked at a functional senescence marker, which is beta-galactosidase activity. We had a positive control called etoposide, which markedly increased from day 2 to day 7. But we also found this strong increase in beta-gal activity between DHX38 positive, crimson positive cells and crimson negative cells, highly suggesting there, though, then that these DHX38 guide RNA effects are acting via cell senescence in these TLO-HEX cells. So we found basically that a knockout of DHX38 induces senescence and senescence-associated secretory phenotype. Sorry, I wrote this wrong here. It's a SASP in TLO-HEX in vitro. Now, to summarize the findings from our screen, we performed a pooled CRISPR screen in TLO-HEX cells that identified 42 variants at 26 CAT loci. We validated 6 out of those 8 effects for candidate variants using single guide experiments. One thing that we learned during doing these pooled screens is that the stimulation context of pooled screens is extremely important. So in our screen, we only looked at TNF-alpha, but you could think of many other things that might be important to actually find effects within these cells. For example, shear stress that is changing within atherosclerosis. So those are some other functional things that one might want to look at that might reveal different loci associated with coronary artery disease. And finally, we found signaling effects such as senescence can actually impact the readout of pooled screens and should really be considered when designing those screens in the future. This work is published since March this year, I believe. So we have lots more analysis within the paper, so feel free to check it out. And if you have any questions, feel free to reach out to me if you can't ask today. And at last, I want to thank the lab of Guillaume Lettre. It was a really cool time that I had there. Unfortunately, my postdoc was kind of cut in the middle by COVID. And so I didn't spend as much time with everybody in the lab that I would have wanted to in person, but it was still an amazing time. Jan was in the lab while I was a postdoc there as well. So I had a really good time there. Everyone was very helpful. And I thank you all for your attention. I'm happy to answer any questions.
Thank you very much, Florian. Great presentation. Okay, so I don't think there are any questions from the audience yet, but I can ask a question. So are you planning to follow up on any of the other sites that you have identified? What are the next steps for your study?
That's an interesting and good question. I mean, I now work on a very different topic, right? Like I work more on myocardial infarction, the downstream effect of coronary artery disease, and much more on the imaging side. But I think, in the future, what I would really be interested in is actually translating these types of screens into single cell CRISPR screens, because these pooled screens really are a huge, effort and require so much cell culturing because you need just these incredible amounts of cells, right? And I just want to show them quickly because I have to thank them. Thierry and Melissa did an amazing job in the lab. Like I didn't do the wet lab for this. It's really all their hard work. And they did an amazing job of doing all these experiments. But I think in terms of variant follow-up, first of all, I would translate them to single cell screens just to reduce cost, time, and effort. And then second of all, I would, instead of following up on variants from this screen, which one can obviously do, I think it'd be interesting to try different stimulation contexts, like I was mentioning, like shear stress, or using just different ways of looking at this. But in terms of follow-up, I think, I would be most interested in seeing in which contexts other variants that are identified actually acting.
Yeah. Let's see, we have time maybe for one more question. So in the context of cardiovascular disease, you know that PCSK9 is really well characterized and has been taken into clinical trials, and there is actually it's been commercialized by a company called Verve. Do you think that there are any hits like that from your screen that um, you would imagine could be translated into the clinic?
That's an interesting question. I think PCSK9, pops up also in genome association studies, but I also believe that there's a lot of rare coding variants in PCSK9, right? And so I think these targets that we can really, where we can learn from rare disease genetics to translate to common disease genetics, I think are less likely to come up in these functional screens where we are targeting like just many loci with smaller effects from GWAS. I think these targets are like more likely to pop up when we do like exome wide studies or burden studies where we really can hone down on these strong effect genes. So I wouldn't expect necessarily the screen or screens like this to identify these targets. Of course, they can if you include low frequency, strong effect variants. But yeah, I think also like a lot of these are already kind of found, like they're low-hanging fruit, right? Because they have very strong effects and they're quite rare. Maybe with the increasing efforts in exome sequencing and genome sequencing and just biobanking, if we can do good phenotyping, like if the community can do a good job of phenotyping these like also rarer phenotypes and more outlier phenotypes, then maybe we can really, land on more strong hits like PCSK9 that enable us to translate this into like the clinic later on. And that would be really exciting.
Okay. So if there are no more questions from the audience, I would like to thank both presenters for those wonderful presentations. Thank the audience for attending. Don't forget that the next month we have a seminar by Megan Buckley from the Findlay Lab at the Crick Institute, our very own Adrini Souza from the Roth Lab at University of Toronto. And if you missed any part of the show of the recording, don't forget we have it on the CMAP YouTube channel. So with that said, thanks everybody, and see you next month.